Hi, my name is Adrian Pappas from 1AucseearSDadh Biosciences, and today I'm going to show you how to run a successful cell adhesion assay on our trademark iCelligence system. To get started, I'm going to show you the equipment. So in order to run the cell adhesion assay, you're going to need some very basic materials associated with the iCelligence system. And here we have, in general, the iCelligence unit already equilibrated inside the incubator. And it's currently in standby mode as indicated by the orange lights, as you can see. And the second major piece of equipment that you will need in association with the iCelligence system is the actual iPad, which contains the software to run your experiment. Now, the first thing that you should do in order to make sure that the iPad is actually connected with the iCelligence system is to make sure that you are connected to the correct Wi-Fi network. And in order to determine which Wi-Fi network you need to connect to, it pretty much corresponds to the serial number associated on the actual unit. And so in this case, it's ending in 131. We are connected. In order to set up your cell adhesion assay with the iCelligence system, no special technical equipment is needed aside from your very standard tissue culture reagents and equipment. So over here on the left-hand side, we have just very basic pipettes, cell tissue culture reservoirs, conical tubes, and your very standard, P20 for your cell counting, and various multi-channel pipettes, as well as their corresponding tips, and your pipettement for general pipetting purposes. In terms of tissue culture reagents that are needed, it's very standard again, like I mentioned, PBS to wash the cells, trypsin to detach the cells, as well as their full media, and depending on your assay, how you like to set up your conditions, you may or may not want to set up your cell adhesion assay in serum-free media. In this case, I have it right here, which is the F12K. Aside from that, you'll just need the very basic E-plate L8s as well, and you should be ready to go. Now, previously we had already established that the iPad is in fact connected to the correct iCelligence station. So now we are ready to actually start the software. And once you start the software, it'll prompt you to log in as a specific user. And it all depends on your preference, whether or not you want to be logged in as an administrator, user 1, or user 2. In this case, we could log in as user 1. From here, you have the option of selecting a new experiment, data analysis, or resume experiment. But since we want to set up our cell adhesion experiment, we clicked the new one. From here, you're given the option of what kind of assay you would like to set up, which in our case is an adhesion. And depending on whether or not you already have a standard protocol, or a user-defined protocol that you have previously saved, you could pick standard protocol for now. The next thing you do, it'll prompt you to make sure that you have the correct steps for your experiment. So the first step, as you can see, is already predefined as well as set. In this case, it's a one sweep, one interval, which is essentially going to be our background measurement. Next thing you do, assuming that your steps are okay, is actually lay out your plates. And here the iCelligence station actually has tube slots for two separate plates, but here in the software, the layout is combined for both plates. So if you are just running one plate, you can select to edit your layout for a single plate. And here you can type in your cell type and so forth, and all the necessary information that you would need to clarify the conditions for your experiment. So let's say in this case A549 cells, and you are ready to go. So depending on your assay conditions or your experimental conditions, you could already have your E-plate L8 pre-coded. You could do the coding yourself, optimizing the certain conditions that you'd like. But in that case, your pre-coding obviously would be done before you set up your actual cell culture experiment. And so in order to obtain the background, assuming you have your plate ready to go with the conditions that you desire, you'll need the cell culture reservoir in general for media. And in this case, we'd like to set it up with serum-free media. And so here, my background measurement will be with serum-free media as well. Now each well, we're going to be putting 180 microliters per well, so we really won't need that much media. Now in general, we recommend for your background measurement to be somewhere on the order of 150 to 200 microliters, depending on your preference and how you want to set up the ultimate volume for your experiment. We recommend definitely do not go below 150 microliters. This is to ensure that the media covers the entire well completely for your background measurement and for all of your measurements, so forth. And so I'm going to be putting 180 microliters. Now, as I mentioned, one of your primary concerns should be whether or not your entire bottom of the well is covered, and you can do that by just performing a visual check. Now, if you do notice that the media is not covering the entire bottom of the well, you could simply tap the side of your plate to ensure that it, in fact, is covering the entire bottom. And so now that we have our media in the plate with no cells and so forth, we are ready to obtain our background measurement. Now you're ready to place the plate inside of the station slot. So, now when you place the plate in there, there should be really no resistance when you engage the device. It should be laying nice and flush, and this is just to make sure that the contact pins are actually in contact with the electrodes on the bottom of the plate. And the green light indicates that it is ready to go and a plate is in fact engaged. Now that we have placed our plate onto the station, we are ready to press start and obtain our background measurement. And as you can see, now that it has recognized the plate is in there, the start button actually turns green, whereas prior to that it was actually grayed out. So from here you can click start, and right now it'll prompt you to name your experiment, and so we can pick the default name, which is associated with, it's essentially the date as well as a certain serial number for the experiment. And so we can just click OK. And the first measurement has been taken. And so just to do a sort of quality control, make sure your plate is working, make sure you have no dead wells and so forth, you can actually add all the wells to look at. And in fact, we are starting at 0. So your first time point should be 0. Now from here, now that we have obtained our background measurement, you're ready to actually add your cells. Then you'll need to take the plate back out from the station. So you just disengage it. There's actually a button in the front that lets you open and close each cradle. And so removing the device should be trivial. So now that you have obtained your background measurement, you're ready to put your cells onto the E-plate. Now you're going to want to resuspend your cells in your desired media at the desired concentration for the plating volume that you're going to apply into the plate, which in our case is going to be 300 microliters. Now we recommend in general to run your assay anywhere between 400 to 600 microliters per well total. So in our case, since we already have 180 microliters for the background, I'm going to be adding 300 microliters of my cells. And so my density is such that 300 microliters equates to the cell density that I would like per well. So in general, just standard tissue culture practice, you're going to want to make sure that your cells are nicely suspended. And so I'll just be pipetting up and down. You want to make sure that's a homogeneous mixture such that each well receives, statistically the same number of cells. Now here, since I'll be adding 300 microliters of cells, I have a 1 ml multi-channel pipette, and in case you don't have it, you can also use a 200 microliters multi-channel pipette and just add twice. Either way works for you as long as your cell mixture is homogeneous enough. Okay, and now that I've added my cells, we're actually going to be equilibrating this plate at room temperature, and that is just essentially to make sure that your cells settle homogeneously onto the bottom of the plate and nice and evenly and so forth, because if you were to take this plate and put it on the station immediately, your cells may settle in aggregates or non-uniformly. In general, for most isologence assays, we actually recommend incubating your plate at room temperature for 30 minutes, but in this case, since we're doing a cell adhesion study, we want to actually monitor those initial settling and attachment kinetics. And so in this case, I actually recommend incubating your plate at room temperature for around 15 to 20 minutes. That way, at least your cells will have settled enough and attached and adhered enough such that when you take your plate and put it on the station, there'll be minimal detachment of your cells. And so I have my timer running and just incubate. Now we have our cells on our plate, and we want to very gently put the plate back onto the station to begin recording. Green light indicates you're ready to go. And you're ready to start your experiment. So as you can see, it's pretty easy to set up the cell adhesion experiment on the isologence system. We went over how to set up initially your isologence unit and make sure it's communicating with the iPad. We went over how to initiate the software, how to get your experiment set up and running with the schedule and the layout and so forth. We went over the background measurement, how to set that up, and which is very important for your initial reading, which your subsequent cell index measurements will be compared and referenced to. We went over the general plating of your cells into the plate as well as the required volumes and so forth. As well as we went over the incubation time at room temperature to ensure initial homogeneous cell seating on the bottom of the plate. And so that's cell adhesion for you. Thank you for watching, and tune into our other videos where I go over the other 3 assays, which include the cell QC, proliferation and viability assay, as well as the receptor ligand assay. I'm Adrian Pappas from 1AucseearSDadh. Thank you very much.